Welcome back! Last time we introduced DNA by describing its structure and the important role it plays in an organism's ability to grow and develop. Tonight, we will begin by describing one of DNA's unique functions, which is its ability to replicate or copy itself. Later this year, we will learn that organisms grow when their own cells make new copies in processes called mitosis and meiosis. So we will learn the details of those two processes later this year, but for now, I want you to think that every time a cell makes a copy of itself, the new copies will need their own set of DNA. Therefore, DNA is going to be replicated before these processes of mitosis and meiosis. So, tonight we are going to go through step by step how DNA can replicate or make copies of itself. So let's begin by looking here at our DNA double helix. This is our original double helix, and we're going to see how we can take this double helix and from it make two. So let's begin by remembering that the DNA double helix is really two strands, two sides of a molecule that are put together by nitrogen bases in the center. These are two different nitrogen bases on each rung that are bonded to each other to keep the two sides of the molecule together. So when we are ready to replicate our DNA molecule, the first thing that must happen is that we need to unzip the two sides of the double helix. Now, if you think about unzipping a jacket, we know that we use a zipper to separate the two sides. In the case of DNA, we are going to use this thing here. This is an enzyme. As you've already learned, enzymes can make or break bonds. Oops, sorry, I'm going to spell this wrong. Let's not spell it wrong. All right, so we've got our enzyme, and in this case, the enzyme is going to be breaking the bonds between the nitrogen bases. So this enzyme here will actually move up the DNA strand, and as it does, it will break apart the bonds between those nitrogen bases. As it does this, the two sides of the ladder are going to be separated from each other. All right, now we have two separated DNA strands. Here's one, and here's the second one. They're now separated from each other, and we can see that these separated DNA strands now have unpaired bases. Okay, so all of these nitrogen bases now no longer have a match. Since they are unattached to their original partner nitrogen base, these unpaired bases can become a pattern on which new nitrogen bases can be added. Now remember that we can't just add any nitrogen bases we want to our separated strands. The new bases we add must be complementary, or in other words, opposite of the original bases. So let's take a look at what we mean. Here, again, we can see our unpaired bases, but what will happen is if we have an unpaired cytosine here, then other free nitrogen bases that are floating around in the cell, like for instance this guanine here that maybe is just floating around the nucleus, can find that cytosine and attach to it. This thymine here then could find this adenine and attach to it, and so on. And as the old strand continues to have new bases reattached to it, we are beginning to build a new second strand to go with our original first strand. Now if we look over here, the same thing is happening over here. We've got the free nitrogen bases on this side being paired up with new nitrogen bases. They're complement or they're opposite. Okay. Until we see that this original second strand, once it gets completely repaired, it's now going to be a double helix again. Okay, so to reiterate, if we had an original strand that had this original sequence of bases in it. So a thymine, guanine, adenine, guanine, cytosine, adenine, thymine, thymine, adenine, adenine, cytosine, guanine, cytosine. Then what would happen is that the cell would have to add a complementary sequence in order to make the molecule double-stranded again. So the complementary sequence is shown here. As you can see, if originally there was a T, then the one that is paired with it has to be an A. You can't add a C or a G, it has to be an A. Next, if the next base in the original strand is a G, then the new one you have to add on has to be a C. And so on down the line. Finally, once all of the unpaired bases on the two original strands are repaired with new bases, the end product is two new identical DNA molecules. So for each of the original strands, here are the two original strands, and if you repair up the unpaired bases on each original strand, you're going to end up with 1, 2 new identical DNA molecules. All right. We have one more topic to discuss tonight before we are ready to talk about how our cells actually use the DNA to make the proteins that determine our traits. We have already mentioned that DNA is an important type of nucleic acid, but it is not the only type. Another nucleic acid needed for making protein is RNA, or ribonucleic acid, RNA. RNA is actually quite similar to DNA because they are both nucleic acids. But there are some important differences between these two types of molecules. First, RNA is a single-stranded molecule. If we compare here, our DNA this side we've got DNA, this side we have RNA. And then we've got, these diagrams are side by side for comparison. So, if we compare our DNA to our RNA, we'll notice that our DNA is a double helix. In other words, we've got not just one strand, but two strands wrapped around each other when we make DNA. However, RNA is basically just a one-sided ladder. It's one strand that kind of wraps around itself. It looks like half of our DNA double helix. All right, second, RNA uses the nitrogen base uracil instead of the nitrogen base thymine. Whenever nitrogen bases in RNA get paired up, which is actually not shown in this picture, but which we will see happen in some processes described in our next lecture, uracil will always pair up with adenine. So let's look again at the comparison diagrams. Next to each nucleic acid, we see the nitrogen bases used to make up that nucleic acid. So here we've got cytosine shown in both. So we've got our cytosine. RNA and DNA both use cytosine. They both use guanine. Okay. They both use adenine. So all the way up till now, the pieces are the same. But at the bottom, you'll see a slightly different nitrogen base. Here, our uracil is going to replace or be used instead of thymine. And if you look at those molecules, they are very similar to each other. There's a small difference in the molecular makeup right there compared to here. Okay, so very slight differences, but not insignificant. And we're going to see this in again in the processes we describe in our next lecture. Okay, finally, the sugar used to make the sugar phosphate backbone in RNA is called ribose, not deoxyribose. So recall that our backbone here is rotating sugar, phosphate, sugar, phosphate, all the way up both sides of the ladder. And remember, for DNA, that sugar was called deoxyribose, which is where the D in DNA comes from. Here, we have the same pattern of sugar, phosphate, sugar, phosphate to build the backbone of the molecule. But in this case, the sugar is just slightly different. It's a ribose instead of a deoxyribose. Okay, great. We are done. Oops, we're not doing that. We're done for tonight. Take a look back at your notes and then go ahead and take your Quizmodo. See you next time.